Thank you for joining CADASIL Association's 5th webinar in our CADASIL Connection speaker series. I am Jane Gunther, and I will be your moderator. Today's speaker is Dr. Michael Wang. He'll be presenting his most recent results from his lab's research using molecular clues from CADASIL brain analysis. Before we begin, I would like to go over a few housekeeping items. We've taken a screenshot of an example of the attendee interface. You should see something that looks like this on your own computer desktop in the upper right corner. You were prompted to select your audio, and the default setting is computer audio. If you prefer to join over the phone, which can provide better audio quality, select telephone in the audio pane, and the dial-in information will be displayed. You have all joined this webinar in listen-only mode. All callers are automatically muted. At the end of the presentation, Dr. Wang will answer questions that were pre-submitted during the webinar registration process. However, at any time during the webinar, we invite you to type questions in the question pane of the control panel. These questions will be forwarded to Dr. Wang following the webinar. And even though these questions will not be answered today, we value your questions, and answers will be posted on our website. Today's presentation is being recorded and will be available on the CADASIL Association website. The devastation that CADASIL had on their families led a group of volunteers in 2012 to establish the CADASIL Association, whose mission is communication, awareness, education, research support, and to act as a patient resource. The dedication and passion of these volunteers to CADASIL has led to a number of accomplishments by our association. This includes our ongoing physician outreach program, which provides awareness and education by supplying physicians' offices with brochures on CADASIL for those families newly diagnosed. In 2016, we hosted patient registry rallies at 4 locations in the U.S., and our newest educational event is CADASIL Care. Our board member, Sandra Talbert, received a Global Genes Rare Patient Impact Grant, and with the support of this grant, CADASIL will be hosting a CADASIL Care speaker event this fall. CADASIL Care is a patient education event dedicated to medical and genetic education topics for CADASIL families in the convenience of 20-minute recordings presented by experts in CADASIL and genetics. We continue to support strong research initiatives, which has included 3 summer research scholarships for students working in U.S. CADASIL labs at the University of Michigan in Dr. Wang's lab, Dr. Arboleda Vesquez's lab at Harvard, and Dr. El-Bajani's lab at Davidson College. We are in the process of awarding a 4th scholarship for this summer, which is continuing our investment in future CADASIL researchers. We are pleased to be in the position to increase our financial support of a research alliance with Dr. Joe Arboleda Velasquez's lab and their work on biomarkers and therapeutic targets. This year, we are sponsoring a student travel award to the 25th Annual American Society of Neurotherapy and Repair Conference. Suning Ping, a graduate student in Dr. Li Ru Zhao's lab at SUNY Upstate Medical University in Syracuse, received our student travel grant to this conference. Suni is presenting today at the conference on her work entitled Stem Cell Factor and Granulocyte Colony Stimulating Factor Promote Brain Repair and Cognitive Function through VEGF-mediated angiogenesis in a mouse model of CADASIL. This award was made possible by donations from CADASIL board members. To support all the work we do, we have several upcoming fundraising events. In May, there is an online auction of family fun items just in time for summer activities. In June, an Albany Empire Arena football event in Albany, New York. And happening now until July is a large family fundraiser that was initiated by the Carney family. Please check our website and Facebook page for more information on all of these events. And thank you to Michael Kennedy and the Carney family for dedicating their time and energy to make these fundraisers happen. We created the CADASIL Connection speaker series as a platform for researchers and clinicians to present their work to the community. It's been exciting to see this series help accelerate research and create collaborations that we hope will make meaningful differences in the lives of those with CADASIL. To date, our presentations have focused on potential therapeutics and biomarkers, disease pathophysiology, CADASIL diagnosis and misdiagnosis, and the ongoing NIH CADASIL Disease Discovery Study. Today's presentation by Dr. Wang will focus on his work that furthers our understanding of how the mutation of the Notch3 gene affects the Notch3 protein, resulting in disease pathology. I'm pleased to introduce Dr. Michael Wang. Dr. Wang trained as a stroke neurologist as well as a molecular biologist. He received his undergraduate degree in chemistry from Harvard College. His MD training was at Johns Hopkins University, as was his Ph.D. in molecular biology and genetics. Dr. Wang's residency in neurology and his postdoctoral fellowship were both at Johns Hopkins University. Dr. Wang is currently Chief of Neurology at the VA Ann Arbor Healthcare System and Professor of Neurology at the University of Michigan, where his lab studies the biology of vascular diseases of the brain, with special emphasis on the pathogenesis of ischemic stroke and inherited vascular dementia, such as CADASIL. Dr. Wang found it a natural fit to use his interest in molecular biology to help us understand the molecular basis of the most common genetic form of stroke, CADASIL. Dr. Wang is an active member of cureCADASIL's Scientific Advisory Board. Dr. Wang's lab has received federal funding to study CADASIL through the VA program and other federal grants for the last 10 years. The CADASIL research in Dr. Wang's lab is focused on the mechanisms of vascular degeneration and on defining the pathological proteins that abnormally build up in CADASIL. Dr. Wang's lab has identified novel vascular proteins that accumulate in CADASIL. Dr. Wang will share his lab's work in advancing our understanding of CADASIL, and then in the second half of the webinar, Dr. Wang will present on the CADASIL Brain Donation Program, which is a collection of CADASIL patient brains. That is a free donation. There's no cost to make this donation, and then there's no cost to interested researchers to obtain samples from this repository. Welcome, Dr. Wang.
Thank you very much, Jane, for the kind introduction. It's really a pleasure to present our work on the molecular pathogenesis of CADASIL in this forum. And I want to thank the CADASIL Association for the invitation to speak in this webinar. So today I want to tell you in about 30 or 40 minutes about one component of our research on the pathogenesis of CADASIL. And this story is highly dependent on analysis of human brains, which we have received as donations in our laboratory in Michigan. And from the outset, I want to express our appreciation for all the generous donations from the CADASIL community. I'd also like to acknowledge members of our laboratory from the onset. These are very talented individuals in my laboratory at the university and at the VA. These are Sujung Lee, Xiaojie Zhang, Name Pearl Carty, and Kelly Young. And here's a picture of our lab to put faces to the names. The four individuals whose work I'll present today are joined by a number of very talented students from all over the country, in particular from the University of Michigan. And we had the great fortune to have one of our University of Michigan students sponsored by the cureCADASIL Association, and we're very appreciative for their generous support. I'd also like to acknowledge some of the federal funding and private funding we've received. We have received in the past grant support from NIH, specifically the National Institute for Neurologic Disorders and Stroke, from the VA, and also, as I mentioned, from the cureCADASIL Association. And we've received very generous donations from CADASIL Together We Have Hope over the last many years. Again, I want to reiterate my strong appreciation to the CADASIL community for their generous donations of tissue and for funding. It is just a wonderful experience to gain support from the community, and it's certainly a great inspiration to all of us in the lab who are working on this problem. So this is a roadmap of where we're going today. We really have two main destinations. The first destination is to describe some of the basic research that we're performing. And I'll spend most of our time talking about one basic research problem that we're trying to solve. And the second part of the talk will touch a little bit upon the brain donation program that Jane described, and hopefully provide a little bit of information as a starting point for conversations for people who are interested in making donations. So I don't need to give too much of an introduction to CADASIL, but it's useful to revisit the definition of this acronym. As all of you know, this is a brain disease, and it's a genetic brain disease that is inherited in an autosomal dominant fashion, meaning that one copy of a defective gene is required to pass this disease on to members of one's family. The disease is an arteriopathy in that it affects the arteries of the body, and in particular, the arteries of the brain. The result of arterial disease is defects in the subcortex of the brain. So these subcortical problems result in infarcts and ultimately in leukoencephalopathy, which is a fancy way of describing white matter disease within the brain. CADASIL is therefore a monogenic disease. It is a monogenic small vessel disease that causes arteriopathy. And the principal clinical problems which face CADASIL patients include stroke, vascular dementia, and psychiatric symptoms, including anxiety and depression. It's useful to revisit the MRI scans of living CADASIL patients. These images show typical findings within CADASIL brains. These include anterior temporal lobe white matter hyperintensities, which you see on the left. And you also see frequently on MRI scanning external capsule hyperintensities. The first breakthrough research finding in CADASIL was the identification in France of many families with this disorder, and ultimately the use of molecular genetics resulted in mapping of the gene responsible for CADASIL. And through a great deal of hard work, Jutel and colleagues in Paris were able to narrow down the molecular cause of CADASIL, which was determined to be mutations in a gene called Notch3. So Notch3 is one of 4 Notch genes. These Notch genes encode proteins that are expressed at the cell surface of cells and are responsible for guiding cell development. A few years later, Jutel described a large number of CADASIL families and the molecular genetic basis of the disease in these families. And as you can see, this is a long list of mutations that were found in many families in Europe. And what's striking about the molecular basis of this disease is that all of the mutations in the Notch3 gene alter cysteine content. So cysteine is one of 20 amino acids, and cysteine is famous for forming disulfide bonds, which are bridges between different parts of proteins. Jutel's group went on in 2000 to describe some striking immunohistological changes within CADASIL brains. And their main finding was that the Notch3 protein accumulates in a very powerful fashion within vessels of the brain. And you can see in this micrograph to the left, there is a rather strong accumulation of Notch3 in CADASIL brains, which is not seen to the same extent in control brains. Western blots suggest that there's a very powerful accumulation of Notch3 protein in CADASIL. So naturally, once the gene had been identified and the protein described in blood vessels of CADASIL patients, the next step was to try to model the disorder in mice. So mice are a very common animal model for human disease, and Jutel made the first attempt at animal modeling in 2003. This was a standard transgenic model using an SM22 promoter, which enabled the overexpression of Notch3 in mice. And as you can see, this was a success in that Notch3 could be overexpressed in mouse tissues. Moreover, accumulations of granular osmophilic material, which is characteristic of human CADASIL, was found in these transgenic mice. So this demonstrated that it was possible to overexpress Notch3 in mutant form in mice. Unfortunately, these mice did not have a neurological phenotype. They did not experience cognitive deterioration or stroke. A number of other models have subsequently been published, and I'm just going to show you a few. A large number of models have been made from our lab, from Joe Arboleta's lab, and a number of other groups. This is Urban Lendel's group, and they made a knock-in model in mice where they introduced a human mutation into the mouse Notch3 gene. What they found was that there was no neurologic phenotype in these mice. Jutel went on in 2010 to create something called a BAC mouse. A BAC is a bacterial artificial chromosome, and Jutel's group fished out a BAC encoding rat Notch3, created a mutation in the rat Notch3 BAC, and introduced that BAC in high copy number into mice. They were able to overexpress Notch3 in these mice. Moreover, expression using a BAC vehicle resulted in more histological phenotypes that were similar to CADASIL, including white matter hypoattenuation and gliosis. Unfortunately, we have not seen reports of neurological phenotypes in these animals. More recently, Wales has published yet another model of CADASIL by introducing mutations in Notch3. And when these mice are aged out to 22 months, which corresponds to about 70 or 80 years in human time, these mice start to develop some histological phenotypes and some motor signs. So at a very low frequency, at a very high age, some animals that carry mutated Notch genes are demonstrating some neurological phenotypes. So the overall result of all of this work in mice is that, yes, we are technically able to generate transgenic mice that overexpress mutant Notch3. We have the technology to do that, and the mice have been generated. Unfortunately, these mice are relatively resistant to mutant Notch3. None of these mice really demonstrate strong pathological changes that represent changes in human tissue, and none of these models demonstrate some of the core findings of CADASIL in humans, which includes stroke and cognitive changes. So I think at this point, although we have not been able to successfully model all of the features of CADASIL in mice, I can say that the field in general has made a lot of substantial gains. First of all, we've identified the mutations and the molecular basis for CADASIL. There is no doubt that Notch3 protein is a central player in this disease. There's also no doubt that mice are resistant to mutant Notch3. For some reason, when we introduce the same protein that's present in human patients with CADASIL into mice, the mice don't mind the protein and don't get sick. So I think this finding has stimulated us to really focus on human tissue. If mice are resistant to mutant Notch3, we've asked what's different about the protein in human tissues. Are there further changes in the human protein that are not found in mice, and are these changes important in the development of the disease? So what I'm going to tell you about is some work that we've done over the last few years to identify a potential molecular framework for how CADASIL may progress. And this is hypothetical. This is based on some new data that we have, and we still have quite a bit of work to try to prove this model. But this is our working hypothesis of how the disease actually may operate within human vessels. And what I'm proposing is that Notch3 undergoes not just a single change introduced by mutations, but must undergo multiple changes over time to adopt conformations that are pathological in people. So in other words, what I'm hypothesizing is that there's a premutation which makes a pre-pathological protein. This mutation, which disrupts a cysteine, breaks one cysteine disulfide bond. But this protein appears to be insufficient to fully recapitulate the disease. What is needed, according to our hypothesis, is further changes in these proteins, which result in multiple disulfide bond reductions and then further conformational changes, which we have yet to define. So let's go through some of the data which support this hypothesis. So the data really goes back to an experiment that did not work. We like to euphemistically say this was an underperforming experiment. In 2004, we decided to make polyclonal antibodies against Notch3. If you're going to study a protein, you need antibodies to be able to quantify the protein and identify where the protein is deposited in tissues. So we took a straightforward approach. We intended to make a polyclonal antipeptide antibody against Notch3. So we targeted a region in the N-terminus of Notch3 and generated two anti-sera 2078 and 2079. And when we tested these antibodies, they failed to recognize Notch. And here's one of the experiments we did. We took cell lines that we knew were expressing large amounts of either wild type or mutant Notch3, and we simply used our antibodies to stain these cells. And a control antibody, a commercial antibody, stains these cells very, very well. However, our antibodies failed miserably to stain Notch3 that was produced recombinantly in cell culture. So the failure did not end there. We tried these antibodies on Western blots, and they failed to bind to Notch3. So we decided we would just freeze this set of antibodies. We put it in a freezer and pretty much forgot about it for about 5 years. In 2009, about 5 years after we had developed the antibodies, we received a call from a patient's family. The patient with CADASIL had died, and the family was very interested in contributing to research, and they decided to donate the CADASIL patient's brain to our research program. And we were very excited to receive this donation, and we thought, well, this is the first CADASIL brain that we've received in the lab. Let's try those antibodies that were in the freezer that we gave up on. Let's just see whether these might work in an immunohistochemical experiment. And we were very surprised after our experiments showing that the antibodies did not work on Western and did not work on cell culture, that the antibodies highlighted the vascular media in a very potent way. To make a long story short, we have applied these antibodies to 18 patient samples. So these are 18 CADASIL brains. And both of these antibodies, which don't work on Western and don't work on proteins expressed in cell culture, both of these antibodies work very well in most tissues with CADASIL. And if we take tissues without CADASIL, the normal looking tissues fail to react with these antibodies. So one theory about why these antibodies were working on tissue, but not on Western blot or on protein expressed in cells, is that maybe there's a conformational change that is going on that these antibodies are detecting that are not present in recombinant protein that we isolate in the lab. And to see whether this concept might be true, we generated other antibodies, again in the N-terminus of the protein, but just adjacent to the original epitope. And to make a long story short, we were able to use these antibodies successfully in immunohistochemical experiments, but these antibodies also exhibited the same property in recombinant protein made in cells and purified from cells. These antibodies failed to bind to 293 cells, which overexpress the Notch3 protein, and also failed on Western blots. But like the previous two antibodies, these antibodies were very effective at identifying diseased vessels within CADASIL tissues. So we have 2 epitopes and 4 polyclonal sera, and all of them exhibit this very interesting property in that they bind to protein within diseased tissue in CADASIL, but do not bind to a protein that's recombinantly produced. So to determine what might be the change that occurs in human tissue that results in antibody binding, we took recombinant Notch3 protein. So this is the same protein that doesn't work on Western blots with these antibodies. And we simply treated these recombinant proteins in vitro with a variety of reducing agents. And we used DTT, BME, and TCEP. TCEP is a non-sulfur-based reducing agent. And we discovered that in a dose-dependent fashion, we could reveal the epitope that this antibody was recognizing using reducing agents. And this was true for both the 5210 and the 2079 antibodies. Both of these N-terminal antibodies that recognize tissue epitopes appear to only recognize Notch3 when Notch3 is reduced. This is not a function of simply unfolding the protein. If you take CADASIL Notch3 protein that's made recombinantly and you beat up the protein by heating it or treating it with detergents or chaotropic agents or alkylating agents, none of these rather harsh treatments result in binding of the antibodies to the protein. It's only the reducing agents that result in binding of these disease probes to the Notch3 protein. So these antibodies indicate that a version of Notch3 that is not the same as what is produced acutely in cell culture, a different version of Notch3 is probably present in CADASIL tissues. We've called this by a number of different names in the lab. This is one of the names, reduced Notch3 or multiple reduced cysteine Notch3. What we're hypothesizing is that in disease tissues, we are not we are seeing more than just single mutant protein. We're seeing protein that is extensively reduced. And it's this extensively reduced protein, we think, that might be driving disease. So we've gone a little bit further. The first few experiments, which we have published already, were using polyclonal antibodies. We have now generated monoclonal antibodies against the N-terminus of the protein. And we've screened the monoclonal antibodies using a number of criteria, ELISA, reactivity with tissues, and then we've scored these monoclonal antibodies for their ability to bind to Notch3 protein under various circumstances. I just want to show you a little bit of data. This is one of the antibodies that we've, this is a monoclonal against Notch3, which we have identified. This monoclonal, when tested against CADASIL tissue, strongly reacts against CADASIL vessels. And if you go to higher power, this reactivity is in the media of the vessels, and that's exactly where Notch3 is found. This same antibody, when applied to normal vessels, does not react with normal media. This is another antibody, 1E7, a monoclonal from rat. And when 1E7 was tested on CADASIL tissue, we saw intense staining in the media of leptomeningeal vessels. This is where Notch3 is deposited. The Notch3 protein is not recognized by 1E7 when the protein is not reduced, but when you take this recombinant protein and you treat it with increasing concentrations of reducing agents, you start to see reactivity. So 1E7, this new monoclonal antibody, behaves very similarly and has a similar specificity to some of the polyclonal antibodies that we have identified. Now, I want to point out in all of these experiments, we have compared wild type to mutant protein, and none of these antibodies preferentially recognize the mutant protein. They recognize both the wild type and the mutant protein, but only when the protein has been treated with reducing agents. This agrees with our hypothesis that Notch3 may be extensively reduced in human tissue. So to go one step further, we've made proteins, recombinant proteins, and some of these recombinant proteins have been mutated in multiple ways. So this is a single mutant protein. We have generated proteins that are mutated in 2 to 5 residues of Notch, all in cysteines, and you can see that this monoclonal antibody reacts with some of these multiply mutated Notch3 clones, but not to the single mutant protein. Again, consistent with the hypothesis that some of these monoclonal antibodies could be recognizing a protein that is extensively reduced rather than reduced in a minimal fashion, which you might expect with an acutely produced single mutant protein. So some of these monoclonal antibodies have properties that are a little bit different from the polyclonal antibodies. For example, some of the monoclonal antibodies bind to tissue but don't bind to any of the protein that's treated under any circumstances, including using reducing agents. And so we've scored the characteristics of these monoclonal antibodies. We've included specific criteria, like whether the recombinant proteins after reduction bind to the protein, or whether these antibodies work on immunohistochemistry of cells versus tissues. And what we ended up was a categorization of different monoclonals. So we could categorize these monoclonals in at least 3 different ways. And these three different groups of monoclonals suggest that there are at least 2 or 3 different conformations of Notch3 that might exist in CADASIL tissue. So because we have found that these antibodies bind to unusual forms of Notch that are not present in normal tissue and are not present in a wild-type protein that is produced under normal circumstances in tissue culture, we suggest that CADASIL may be a proteinopathy. It's a disease that is based on pathology of specific proteins. And this is very consistent with the molecular genetics, which Jutel very nicely laid out many years ago, suggesting that cysteine mutations are required for the disease. Cysteines, as I mentioned before, are very important in the secondary structure of proteins, for example, in BPTI and EGF. If you disrupt any cysteine, the protein unfolds and becomes dysfunctional. You might expect that because Notch3 is a disease that requires cysteine mutations, that the protein Notch3 may be deformed in CADASIL. This is not a new concept in neurologic diseases. Other diseases I'd like to point out are thought to be proteinopathies. These diseases feature specific proteins that are misfolded, and many of these will be recognizable by the audience. Alzheimer's is perhaps the most famous neurological proteinopathy, and mutations in or malformations in A beta and tau are really the hallmark features of Alzheimer's disease. The prion disease, Jakob-Creutzfeldt disease, is caused by conformational changes in the prion protein, and Parkinson's disease is associated with malformations of a number of different proteins, including synuclein. What I suggest, and what others in the field have suggested, is that CADASIL may also be a proteinopathy, but the proteinopathy is caused by the conformational changes in a different protein, specifically Notch3. So what I've shown you is some antibody-based data that suggests that there are alternative conformations of Notch3 that are present in CADASIL tissues, but are not present in protein that's produced in the lab. And what we know is that CADASIL requires the mutation of one cysteine residue. I think to fill in the blanks, we need to find out what kind of factors may result in the transformation of a single mutant protein into these multiply reduced proteins. And I should point out, now we have probes against these different forms, and we're trying to characterize what the differences are between these different probes in the different forms of Notch3, and importantly, what factors, such as redox stress or matrix stressors or protein aging, might facilitate the conversion of these proteins into these deformed Notch3 molecules. I'd also like to point out, going back to the question of why mice are resistant to mutant Notch3, I'd also like to point out that we've looked for these conformations of Notch3 in many Notch3 transgenic mice, and we have not been able to find a significant amount of expression of these advanced conformationally changed Notch3 molecules. So this is another representation of what we think is happening. There is probably a redox-dependent change in Notch3, which results in a strongly altered Notch3 protein, which is, we suspect, more pathological than an early protein that has a single mutation. So what does this mean in terms of potential therapeutics? Well, the first thing that pops to mind is that we may be able to target this protein. This strongly altered protein could be a therapeutic target. If we can design a way to eliminate or clear this strongly conformationally altered protein, maybe we have a chance at altering the course of the disease. Another potential therapeutic target is to first understand what causes this transformation from a well-behaved notch to a very conformationally changed notch, and then target the processes that are involved that modulate this transformation. So the future directions are many, and one of the directions that I would like to suggest is that the antibodies that we have developed could be used therapeutically. So we know that these antibodies target strongly conformationally changed Notch 3. Might these antibodies be useful in enhancing the clearance of the pathological protein? This is food for thought. We don't have antibodies that are humanized yet, but we've made the first step in developing some reagents that might be useful. So that's the end of our main part of our talk. And as I mentioned before, and as Jane mentioned in her introduction, I just want to say a little bit about our CADASIL brain donation program that we have in Michigan. It was started in 2009, and we've been very grateful to have met with a lot of success thanks to the collaboration of the CADASIL community. This is just to remind me to tell you about the importance of studying human brains. Right now, our mouse models are not fully recapitulating the human disease. So we have to go back to human tissue and try to find out why these mice are resistant to disease. And the best way to do that is to really go into depth and to understand what's going on in human tissue. So here are some overview facts of our brain resource. It's composed of brains that have been donated by patients and patients' families who have CADASIL. We have in our repository a number of different forms of tissue. We have frozen tissue, and we also have fixed tissue. This should be fixed, which is tissue that is preserved in formaldehyde. And both of these types of tissue are useful in different ways. All of the tissues have been genetically characterized. The tissues that we have been studying have all been genotyped and have characteristic Notch3 mutations. We also have cross-sampled from other brain banks. So we've received samples from Europe and also from other brain banks in the United States, and have added that to our collection so we can compare not just tissues that we have sourced at Michigan, but also tissues that have been donated to other centers. So this is a representation of the genotypes of the CADASIL brains that are in our collection. The lighter blue bars represent individual brains that are in our collection. The darker bars are living patients we follow in Michigan, and these are the locations of the mutations within the Notch3 ectodomain. You can see that a large portion of our brains contain mutations in the N-terminus of the Notch3 gene. So how do you make a brain donation? It's pretty simple. Please contact me directly. It's very easy to contact me. The information is on our website. Feel free to email me, and we can also have a telephone conversation. It's best to plan well ahead. I know a number of individuals around the country have already talked to me, and you guys are probably decades ahead of the game, but it's better to be prepared than to try to piece things together at the very last second. The brain donations, as Jane mentioned, are funded by our lab. So we take care of all the costs involved in the brain donation, and there is no added cost to families. And many of the brain donation costs are supported by our grants, and some of the costs have been supported by generous donations from other families who have believed that our brain donation program should be expanded. The brain donations have already been used, and the tissues have been used to for federally funded CADASIL research and to fund experiments that have been already published. So with that, I would like to close. Again, I'd like to express my great appreciation for cureCADASIL for giving us the opportunity to present our work. And again, I want to thank the CADASIL community for their unending and inspiring support.
Thank you, Dr. Wang. It was very interesting, your research, and really it's some great progress. I look forward to seeing where you take this in the future. We're going to take a few questions now that were pre-submitted to us during the registration period. And our first question came from as I mentioned, from the pre-registration period. Are there any benefits of medicinal marijuana for prevention or recovery before or after stroke?
So I believe this is a question about before and after stroke in CADASIL patients. And I am very interested in that question. That's a great question. I did not know the answer to the question, and I went to PubMed to see if I could find any answers. And unfortunately, I don't see any literature supporting the use of marijuana in CADASIL. So I don't think there's enough information at this point to say anything about that.
Okay. Is there any correlation between CADASIL and an elevated D-dimer blood test?
So this is a good question also. A D-dimer test is used to indicate whether a patient might have blood clotting. And I do not know of any reports of a link between CADASIL and elevated D-dimer. CADASIL is not thought to be due to clotting of blood vessels in the brain. Rather, it's a degeneration of the blood vessels which affect blood flow regulation. So we wouldn't expect the D-dimer to be elevated in CADASIL. CADASIL patients, if they become immobilized and don't have good circulation to the legs because they're not moving as much as other people, may in fact have blood clots in their legs, which may result in an elevated D-dimer. But there's no direct link between CADASIL and D-dimer.
Okay. All right. We have a couple more questions here. To your knowledge, are there other animal models of CADASIL besides mouse models?
So that's another great question. We've just reviewed many of the mouse models that are available in the literature, and we reviewed the fact that many of these models don't develop neurological phenotypes. So there's really a need to find animal models that have some of the neurological features of human CADASIL. To my knowledge, there are no models besides the mouse models that have been developed in vertebrates. I think several groups around the world are interested in developing alternative animal models. None of those have been published to my knowledge. We are actively trying to improve upon mouse modeling in our lab, and our studies are in preliminary stage, but I hope to be able to share the results with the group perhaps in a later webinar.
That would be terrific. I look forward to that. And our last question is, we've heard of CADASIL and CADASIL. Is CADASIL a recessive form of CADASIL, or does it affect a different gene other than Notch3?
Right. So CADASIL and CADASIL share some features. Both are small vessel diseases that affect the brain. Both are monogenic disorders. They come from problems in a single gene. The CADASIL, which is a recessive form of small vessel disease, is distinct clinically from CADASIL. It's a different disorder, and it is a result of mutations in a different gene. So the pathways that result in CADASIL are likely to be different from the molecular pathways that result in CADASIL.
Okay, thank you. I'm going to ask you a question that we did not discuss previously, but looking at the results of your data, you spoke of targeting the process that results in this reduced protein in the human. Are you aware of any drugs that, I know you don't know the mechanism yet, but you might have some hunches as to what that might be, and if so, are there drugs that are already out there that could target that transformation? Or are you looking at more of an antibody approach?
We're looking at both of those approaches. We don't really have any specific targets for that transformative process. My hypothesis is, or I should say my guess is that a reductive process might be involved. So a redox-based process might drive the continued transformation of the protein into a more conformationally challenged molecule. There are multiple pathways that regulate redox balance within the cells, and it would be wonderful if a specific pathway were involved in the transformation of Notch to a highly conformationally challenged protein. If it's a single protein or single redox system that's involved, that could potentially be targeted without too many off-target effects.
Thank you for taking that question. Okay, well, in closing, our goal has been to connect the CADASIL community. So please contact info@curecadasil.org if you'd like more information from our speakers or if you have a topic of interest. One suggestion we have received from our audience is to announce our volunteer opportunities. This is a short list of our immediate needs. Please contact us, and we look forward to meeting and working with more people in the CADASIL community. So in closing, our next CADASIL Connection speaker webinar is going to be in November. We're taking a break over the summer, and we hope that you can join us back in November. In addition, visit our website to learn more about our CADASIL Care speaker events that will be taking place in the fall. Recording of today's webinar will be available at our website. Thank you to Dr. Wang and his lab for their dedication and excellence in CADASIL research. Thank you to our audience for your participation, and this is the conclusion of our webinar.